So, dear attendees, we are very pleased to welcome you today for our second webinar on cyclodextrins, organized together with Rocket and CarboHyde. The first one took place in June 21st, dealing with cyclodextrins in biopharma. Today, we will deep dive in cyclodextrins in pharmaceutical applications, from manufacturing, properties, safety, and regulatory aspects, as well as the next generation of cyclodextrins as therapeutics. 3 presentations will be given today. Each presentation will last about 25 minutes with 5 minutes for questions at the end. After the presentation session, we will go into panel session with our guest, Professor Thorstein Lofstone and Professor R.M. Bilansoy. So the question of today is how cyclodextrins are reshaping the future of pharmaceuticals. During the presentation session, do not hesitate to type in your question in the Q&A section and our presenters will respond to your question after the presentation or during the panel session. We will start now with a first session of 2 presentations titled General Considerations on Cyclodextrins, Manufacturing, Challenges and Properties. So I'm glad to welcome Julien Parcq, our first speaker for today. Julien manages the functionalization laboratory of starches, sugars, and polyol at Rocket R&D. And he will present aspects regarding cyclodextrin manufacturing and challenges. So, Julien, the floor is yours.
Good afternoon, everyone, and thank you very much for offering me this opportunity to share with you some general consideration on cyclodextrin, and more especially on the manufacturing and the challenges associated to that manufacturing processes. So let's review together some general consideration on cyclodextrin. Then we will move to modification of those cyclodextrin before finishing with some key takeaways and some challenges on the production of those cyclodextrin. So cyclodextrin, as you all know, are cyclic polymers, cyclic polysaccharides made of glucose units. They get a conical truncated shape with a small primary side and a wider secondary side. They also have a apolar cavity being hydrophobic and being able to complex with organic molecules. Those cyclodextrin have been identified in the late 19th century by Villiers after he fermented some potato starch with Bacillus acidobacter. Of course, at this stage, the name cyclodextrin was not used, but it was really the starting point from all the further development to come and that you all know right now. There are many available cyclodextrins. The most famous ones are the alpha, beta, or gamma cyclodextrin, all differing from each other with the number of glucose units that they are having or that compose their cycles. Of course, the more glucose unit you get, the larger is your ring and the wider is your cavity. As cyclodextrin are able, thanks to a reversal complexation reaction to complex organic molecules, you easily understand that this wide variety of affordable cyclodextrin offers a wide variety of applications. And really, when you think about cyclodextrin, you think about pharmaceutical application, but you can find cyclodextrin in all your surrounding and your day-to-day life through the cleaning materials, through your textiles, through the cosmetic and hygiene product that you use, or even in your food. Production of cyclodextrin is a well-known and old process, starting with starch mainly from potato that is put in reaction with CGTAs that will be able to cyclase the right chain length to get the different cyclodextrin, alpha, beta, gamma, or other in your reaction mixture. One of the most famous process is the one using organic molecules to complex one of those cyclodextrin and make the equilibrium in favor of the expected cyclodextrin. For example, with beta-cyclodextrin, the use of trichloroethylene or toluene afforded the formation of a complex with the beta-cyclodextrin that helps recovering this beta-cyclodextrin complex out of the mixture. And thanks to the removal of the organic molecule, you can then obtain your cyclodextrin that just need to be purified and that need to be put in the right form or appropriate form. The complexing agent, of course, is reused and is reintegrated in your process. More recently, apart from that process, Sophie Beren's team showed that putting in a reaction some glucan donors with CGTAs may afford the access to a combinatorial library of cyclodextrin that you can really select in regard with your complexing agent, affording the access to alpha, beta, gamma, but even delta cyclodextrin. They even designed a dedicated ligand that complexes with delta cyclodextrin, so they were able to recover delta cyclodextrin complex and really characterize these delta cyclodextrin units. They moved a little bit forward using modified cyclodextrin as a source of glucose for the generation of this combinatorial library of cyclodextrin, affording not only raw cyclodextrin but also modified cyclodextrin that you can access thanks to the complexation with an expected organic molecule that will give you in the end a modified cyclodextrin. Really interesting word that I just advise you to read on. If we consider modification of cyclodextrin, let's focus a little bit on hydroxypropyl cyclodextrin. So hydroxypropyl cyclodextrin are known for a long time. The aim of this hydroxypropylation is to increase the properties of the cyclodextrin, and more especially if we are talking about beta cyclodextrin, to increase the solubility from, let's say, nearly 2% of solubility for the pure beta cyclodextrin up to 65% in water when we are talking about hydroxypropylated cyclodextrin. The process uses, of course, a reactive agent, propylene oxide, that is really reactive and also that possesses some carcinogenic, mutagenic, and reprotoxic properties, meaning that we have really to take care of the handling of such reagent when you put it in reaction. The reaction takes place in water in the presence of an alkaline reagent. And during your reaction, you create a lot of different modified entities from 1 to more than 10 modified structures. It is well known that the substitution at the C2 is favored at low alkaline concentration. A C2 position is the more acidic reactive group, C6 being the more accessible one. So really playing with the alkalinity of your mixture will drive the positioning for the substitution. There is no big impact. It has been shown in the literature that there is no big impact on the substitution pattern for beta-cyclodextrin in regard with the alkaline reagent that you use, as long as you stay with low alkaline or high alkaline concentration. What impacts the cyclodextrin substitution is the cyclodextrin that you use as a raw material. Alpha cyclodextrin tends to form more C6 substitution than beta and gamma cyclodextrin. So your process, as I already mentioned, is taking place in water, putting in water the beta cyclodextrin for solubilization, thanks to the addition of alkaline reagent, in general sodium hydroxide. Then you add your reagent, propylene oxide, and you use the appropriate reaction condition of temperature, pressure, reaction time before you end your reaction, neutralize your mixture, and remove the, let's say, the solvent and the side product to access your expected hydroxypropyl cyclodextrin with the expected form. And then come a real key point is the analysis of all your products. The analysis is crucial because it will then be the characterization of your product, but not only your product, also let's say the side product. So you will first have to determine your degree of substitution, but also the residual beta cyclodextrin, for example, content, residual organics, water content, and all the microbacterial content that could be due to your process. If we think about another modification, we can talk about methyl cyclodextrin. Methyl cyclodextrin consists in putting methyl groups on the hydroxyl part of the cyclodextrin. This is again done with the methylating agent in alkaline conditions using an organic solvent or water. Most famous methylating agents are dimethyl sulfate or methyl halide that again here, as for HP beta cyclodextrin, are toxic reagent, meaning really safety cautions to be taken for the handling of such reagents. Here again, the reaction parameter will play a crucial role, time, temperature in the substitution level and substitution pattern. Because as for HPBCD, alkaline agents drives the reagential activity. You can here again end up with a wide variety of product. If we are talking about the level of substitution, you can have low or high level of substitution or in between level of substitution. You get a high number of substituent that you can get. So really high number of pattern of substitution, 2, 3, 6, 2, 3, 3, 6, 2, 6, and mixture of them. So you see that there are a lot of isomer and regioisomer that you will have to characterize here again when you are talking about methyl cyclodextrins. Apart from those two, there are some really interesting other cyclodextrin that came later on the market. And that's, for example, the example of the sulfobutylated beta cyclodextrin. It is interesting to read in this article from Stella and Rajewski that they really focus on an alternative to co-solvent or surfactant in order to, let's say, solubilize the poorly water-soluble drugs, which is really one of the key points and the key challenge for the use of or the development of new cyclodextrin. So here again, cyclodextrin are put in reaction with the reactive component in alkaline condition in order to afford the desired molecule. There is, of course, a wide range of accessible modification for cyclodextrin. If we talk about beta cyclodextrin, you can talk about non-ionic, anionic, obtain cationic cyclodextrin. You can increase the size of your ring and use gamma cyclodextrin like Sugammadex. You can even consider forming cyclodextrin polymers. So you get a wide variety of capacity to improve the properties of your cyclodextrin. Now, of course, you understand that the main challenge, if you try to produce cyclodextrin, is to increase the properties of your cyclodextrin. So we talk about this in regard with solubility for beta cyclodextrin, that you were able to raise up from nearly 2% to nearly 65% for HPBCD in water. But you also are trying to increase the capacity to solubilize less and less soluble molecules that are in development for the futures of drug development. We are facing more and more class 2 and class 4 drug candidates that will be harder and harder to solubilize, and cyclodextrin, thanks to the capacity to form different kind of complex, may be a useful tool in the future to help in the solubilization of such novel molecules. As soon as you identify these properties, at lab scale, you have to upscale it, and that's one of the challenge that you have to face. How can I generate the molecule with a larger amount? How can I manage my process, and how can I determine my parameters in order to make sure that the temperature, the pressure, and reagent ratios leads to the expected product? How can I manage the handling of a toxic reagent from lab scale to larger scale? And how can I assure the reproducibility of my process? All those are the challenges that you face as a cyclodextrin manufacturer. And finally, of course, the characterization of the cyclodextrin is really the key point. We have to develop the analytical tools to fully identify the substance, master the substitution level, substitution profile, identify the residual impurity, and quantify them in order to comply with the international quality guideline. Lots of hurdles as a cyclodextrin manufacturer in regard with the purity, the identity, and the quality, and also the batch-to-batch reproducibility of the products. So thanks a lot for your attention, and have a nice afternoon.
Thank you very much, Julien. To continue now on the general considerations on cyclodextrins, I'm glad to welcome Dr. Carmen Popescu. Carmen is a global technical developer at Rocket with an extensive knowledge on pharmaceutical technologies and a particular liking for cyclodextrins. Carmen will talk today about the properties of cyclodextrin as a reliable solubilization tool. So Carmen, please take the lead.
Can cyclodextrins be reliable solubilization tools for BCS class 2 and 4 drugs? During this presentation, I would like to draw your attention to the fact that cyclodextrins are present in the composition of many commercially available pharmaceutical drug products. Also, the fact that there are few essential properties an API has to possess in order to complexate with cyclodextrins. Through two case studies, I am going to illustrate the benefits of solubilization of insoluble APIs. Also, at the end, I'm trying to give an answer to the eternal question, what is happening when the API cyclodextrin complex reaches cell membrane. As we can see in these tables, there are a lot of commercially drug products formulated with alpha, beta, and gamma native cyclodextrins. Due to their increased intrinsic water solubility, cyclodextrin derivatives are more versatile, offering access to different routes of administration like oral, parenteral, ophthalmic, nasal. There are few requirements for an API in order to be a candidate for cyclodextrin complexation. The drug molecule skeleton should contain more than 5 atoms, less than 5 condensed rings, water solubility less than 10 milligram per ml, but even at 10 microgram per ml, you still can solubilize an API. melting point below 250 Celsius, molecular weight between 100 and 400, but I was able to formulate up to 1000 molecular weight APIs. It is very important to know if the drug is carrying a charge or is neutral, to know the log P, if the drug has a pKa, or if it has some chemical or photostability issues. If we have a drug candidate with a high log P, high melting point, and it will be administered parenterally at high dose, and it has a suitable molecular shape, we can say, just based on these properties, that this drug is a good candidate to be solubilized by cyclodextrin inclusion complexes. Native cyclodextrins have low intrinsic water solubility. Looking to this example of native beta-CD, its water solubility in comparison with the sulfobutyl ether cyclodextrin, hydroxypropyl beta cyclodextrin, we can see that the derivatives of beta-CD have 25 times higher intrinsic water solubility. That is making them more versatile, more useful in solubilization of higher amount of APIs.
Let's find out how we can increase cholecalciferol solubility by complexation with hydroxypropyl beta-cyclodextrin and determine its phase solubility diagram. What are cyclodextrins? Cyclodextrins are cyclic oligosaccharides derived from starch, with a torus-shaped ring with a polar hydrophilic outside and an apolar hydrophobic cavity. This unique structure allows for the formation of inclusion complexes where lipophilic compounds are non-covalently bound within the cavity. Why cyclodextrins? Enhance bioavailability, reduce irritation, simplify handling, improve patient compliance, stabilize actives, to convert liquid drugs to amorphous powders. What do you need to know about your API to successfully complexate it with cyclodextrins? What is phase solubility? Phase solubility is depicting API solubility increase in presence of different molality of cyclodextrins in supersaturation condition as per Higuchi and Connors. What profile the phase solubility curve can be? B shape, AL shape indicating 1-to-1 complex formation, AP shape. The objective of the study is to increase cholecalciferol solubility by complexation with hydroxypropyl beta-cyclodextrin, HPBCD, and to define its phase solubility diagram. Cholecalciferol, in layman's terms, is known as vitamin D3, a fat-soluble vitamin which is practically insoluble in water at 13 micrograms per liter. Hydroxypropyl beta-cyclodextrins, kleptos HPB, has a molar substitution between 0.58 and 0.68. Vitamin D3's lower water solubility can limit its use in injectable dosage forms. Phase solubility diagram shows vitamin D3 solubility increased at different HPBCD molarities as a linear regression indicating an AL complexation 1-to-1 profile of vitamin D3. HPBCD complexation. Vitamin D3 solubility is increasing by 8 order of magnitude in presence of 200 millimolar HPBCD compared to its solubility in water. Lyophilized samples were analyzed by FTIR, XRD, and DSC to confirm the vitamin D3 HPBCD complex formation, respectively. Vitamin D3 HPBCD complexes are not stable in liquid phase and can be stabilized in a solid dispersion by spray drying. Depending on vitamin D3 polar functional group interaction with HPBCD molecular dynamic simulation is revealing two interaction modes, upstate and downstate. Let's assess the use of beta-cyclodextrin derivatives to enhance grizzle-fulvin solubility by complex formation. What are cyclodextrins? Cyclodextrins are cyclic oligosaccharides derived from starch, with a torus-shaped ring with a polar hydrophilic outside and an apolar hydrophobic cavity. This unique structure allows for the formation of inclusion complexes, where lipophilic compounds are non-covalently bound within the cavity. Why cyclodextrins? To increase aqueous solubility of drugs, to increase chemical and physical stability of drugs, to reduce toxicity, to enhance drug delivery, to mask bad taste or odor, to convert liquid drugs to amorphous powders. What do you need to know about your API to successfully complexate it with cyclodextrins? What is phase solubility? Phase solubility is API solubility increase in presence of different molality of cyclodextrin in supersaturation condition, as per Higuchi and Connors. What profile the phase solubility curve can be? B-shape, formation of complexes with limited solubility in the aqueous medium. AL shape, indicating one-to-one complex formation. AP shape, indicating complex formation of different stoichiometry, starting as one-to-one at the beginning, deviating from linearity to one-to-two or complexes of non-inclusion like aggregates, micelle. Phase solubility study was performed according to the method reported by Higuchi and Connors. Materials: micronized USP-grade grizzle-fulvin and 3 cyclodextrins, kleptos HPB, HP, methyl beta-cyclodextrin from Droquette. Grizzle-fulvin is an antifungal antibiotic used in the treatment of dermatophyte and ringworm infections. It is insoluble in water and is classified as a BCS Class II drug with limited oral absorption due to its poor water solubility. Grizzle-fulvin phase solubility is following a linear regression for all 3 cyclodextrins with the highest complexation efficiency for Droquette methyl beta-cyclodextrin. Higher stability constant and complexation efficiency was obtained for Droquette methyl beta-cyclodextrin compared with HPBCD and HPCD. At 200 millimolar HPBCD, grizzle-fulvin solubility is increasing by 10 times in comparison with its water solubility. Differential scanning calorimetry analysis confirmed the complex formation for all 3 cyclodextrins. In conclusion of the study, grizzle-fulvin solubility enhancement by cyclodextrin derivatives are giving flexibility in selecting the one that is more efficient for the formulation purpose and route of administration. If you are innovating in this space, please do not hesitate to contact Droquette, the global leader in plant-based ingredients, a pioneer of plant proteins, and a leading provider of pharmaceutical excipients.
Let's see what events take place when API cyclodextrin complex reaches membrane phospholipid bilayer. We evaluated using molecular dynamic simulation BCS class 1, 3, and 4 APIs, but due to time constraint, I'll focus only on hydrochlorothiazide, which is a BCS class 4 API. In order to better understand the interaction of hydrochlorothiazide complex with membrane phospholipid bilayer, we partner with Insilibio, a company specialized in molecular dynamics simulation at microsecond time scale. Looking to few time snapshots, we can conclude that hydrochlorothiazide is released from the complex with beta-CD. It stays close to the phospholipid polar head and interacts with carbonyl functional group and water. Thank you very much for your kind and quiet attention.
Thank you very much, Carmen, for your nice presentation. We have time for a very quick question for Julien. So, Julien, from your opinion, what is the main challenge to produce a novel modified cyclodextrin?
On my side, I would say that there are several challenges. One of the most important is the capacity to really handle the process parameters and make sure we get the appropriate molecule in the end. Being able also to make sure we don't create a lot of impurity. So that's another aspect, being the analytical part, being able to identify and quantify the molecule that you are producing, because we are always producing mixtures of cyclodextrin at this stage. The others would be the quality, of course, because we are talking about pharmaceutical application in general. So all these come to this point too. But really the process is an important parameter for me.
Thank you, Julien. Now time has come to start the second presentation, titled Regulatory and Safety Aspects of Cyclodextrin. For this, I am glad to welcome Dr. Damien Truffin, who is a pharmacologist and toxicologist expert at Rocket R&D. So, Damien, please, the floor is yours.
Hello, my name is Damien Truffin, and I'm toxicology and pharmacology expert in Rocket R&D in Nutrition and Health Department. And today I would like to present the regulatory and safety aspects of cyclodextrins, which are important points to consider when developing new pharma applications. In this webinar, we will see a review of literature data on the safety of different cyclodextrins in order to have a global view of the knowledge on safety of cyclodextrin. We will start with the references and sources used for this presentation. Then we will discuss about the oral route of administration with the absorption, excretion, the general toxicity and the carcinogenicity of cyclodextrins. We will also look at the parenteral uses with the pharmacokinetics routes and the systemic toxicity. Furthermore, we will focus on the other routes of administration, ophthalmic, rectal, dermal, and nasal. In addition, we will present the genotoxic, mutagen, and developmental aspects. And finally, we will discuss some regulatory aspects. Concerning the sources and reference used for this webinar, most of the data comes from published papers and especially literature reviews, Gould & Scott, Stella and Hay, and the European Regulatory Agency Q&A published in 2017. This EMA report gives the main information concerning the safety of cyclodextrins used as excipients in medicinal products for human use. Informations are based on the preclinical data described in the marketing authorization dossier of marketed medicines in Europe. It gives a frame concerning the different cyclodextrin families, their usage, and routes of administration regarding their elimination or observed adverse effects, or regarding available data. For example, HPBCD can be used for all routes of administration, excepted nasal routes. Beta-CD cannot be used for nasal and parenteral uses due to its too much impact at renal level. On the other hand, some cyclodextrins have limited possibilities of administration, such as alpha-CD, which can only be used parenterally. Now concerning the oral safety of use and what about absorption and excretion of cyclodextrin. The oral bioavailability of cyclodextrin is very low, no more than few percent in human or animal. And concerning the excretion following oral administration, cyclodextrin are mainly excreted via feces with a low digestibility. If we look more in detail at the different bioavailability value, we can see that the size of cyclodextrin and the chemical nature of derivatives impact the intestinal passage. Moreover, it is important to note that the degree of substitution impacts the percentage of bioavailability. For example, the absorption of methylated cyclodextrin derivative in rats had been reported at 6 to 10% for dimethyl beta-CD and from 0.5 to 11% for monomethyl forms. Besides, cyclodextrins show no interactions with vitamins and other nutrients. The only adverse effects occur at very high dose, more than 1000 mg per kilo per day, with reversible diarrhea and cecal enlargement in animals and also in humans to some small extent. Nevertheless, these effects on gastrointestinal tracts were similar to those of low digestible carbohydrates such as lactose and other widely used pharmaceutical excipients. And its physiological adaptive response to a large load of poorly digestible carbohydrates and other osmotically active nutrients, and the relevance to humans is minimal. Thus, the doses without observed effects or without adverse effects are generally quite high. For example, for sulfobutylated cyclodextrin, the no observed effect level value in dog or in rats after 3 months daily exposure are greater than 3 grams per kilogram of weight per day. And this corresponds for a person of 60 kilograms to more than 200 grams per day. Another example, in dog, after 1 year of orally daily exposure to 1 gram of HPBCD, no effects were observed. It took 2 grams per day for a year to see known adverse and reversible effects in rat and dog. And the major non-adverse observed effects were essentially some impact on urinary tract, epithelial cells, and some impact at pancreas level for rat. In the same way that animals, HPBCD in humans is well tolerated orally with high value of no observed effect level. Indeed, in human, HPBCD was considered to be non-toxic for oral route if the daily dose is lower than 16 grams and well tolerated and safe in the majority of patients receiving HPBCD at daily oral doses of 4 or 8 grams. In addition, for children under 2 years old, up to 200 mg per kilo per day for 2 weeks of HPBCD were well tolerated and considered safe when used in oral solution of itraconazole. Moreover, some native cyclodextrin like alpha, beta, and gamma CDs are accepted as food additive and generally recognized as safe by FDA, and beta CD is approved in Europe as food additive. Now, concerning the carcinogenicity. Beta-cyclodextrin administered orally was non-carcinogen in rat and mice. HPBCD was non-carcinogen in mice at dose up to 5 grams per kilo per day. Nevertheless, in a rat, a study showed slight increase in polypoid tumors of the large intestine at the highest tested dose and tumors of the exocrine pancreas. However, it's accepted by experts that these are artifacts. Indeed, in intestine, similar observations of colonic mucosal hyperplasia have been made in rats after oral administration of raw starches, caramels, sugar alcohols, and lactose. And this effect was due to an increase of osmotic activity of the intestinal contents. And in pancreas, in the rat, HPBCD complexing bile acids and leads to an increase in cholecystokinin, who act as a mitogen, and finally induce cellular proliferation of acinar cells of the exocrine pancreas. Moreover, if you regard mice, primates, and human, cholecystokinin has no mitogenic effect, and therefore these pancreatic tumors are considered a rat-specific effect with low clinical relevance. Now, concerning the parenteral safety of use and the pharmacokinetics and systemic toxicity of cyclodextrin, different studies show that cyclodextrin disappear rapidly from systemic circulation and are renally excreted intact. And in all animal species, the steady-state volume of distribution was equal to extracellular fluid volume. That means that cyclodextrin distributes mainly in the extracellular compartment with no deep compartment or no storage. Moreover, the total plasma clearance for cyclodextrin in all species were similar to the glomerular filtration rate. Approximately 100% of dose was recovered in the urine 6 to 12 hours after intravenous administration. That means that the elimination of cyclodextrin strongly depends on renal clearance, and cyclodextrin disappears quickly after first several hours of administration. Furthermore, cyclodextrins distribute to various tissues, including essentially kidney, urinary bladder, liver, adrenal gland. However, methylated cyclodextrins has a larger steady-state volume of distribution and longer plasma half-life compared to other cyclodextrin derivatives. And this was due to interaction with cellular membranes and hemolytic activities of methylated cyclodextrin. And finally, retention of methylated cyclodextrin in kidney was longer than other cyclodextrin in the renal cortex. Now, concerning the systemic toxicity, some cyclodextrins such as alpha, beta, or randomly methylated cyclodextrin show renal toxicity after parenteral administration, and this makes them not suitable for intravenous route. Conversely, HPBCD and sulfobutylated cyclodextrin can cause reversible effect on the kidney at higher dose. They can induce vacuolation of the kidney tubular cells without loss of kidney function in animals. And if we regard the no effect level values for HPBCD and sulfobutylated cyclodextrin, they were relatively high. For example, for HPBCD, we have no effect level at 50 mg for rat, 100 mg for dog, and 200 mg per kilo per day for monkey after 3 months daily exposure. And in human, dosage up to 1 gram with intravenous route induced no adverse effects. Moreover, 250 mg per kilo per day was found to be safe in humans older than 2 years when given 21 days HPBCD or 6 months SBOCD intravenous route. So, HPBCD and sulfobutylated derivatives induced no major impact in humans using IV routes. They are considered safe at relatively high doses and used most widely in parenteral products. Another point concerning the usage in children, because of their lower renal function, children less than 2 years old may theoretically be less vulnerable to renal toxicity than other cyclodextrins, with high HPBCD blood levels due to slower elimination. Moreover, in juvenile rats, toxicological effects of HPBCD were no worse than in adult rats. Indeed, a study performed in juvenile rats during 44 days, the authors show that HPBCD was generally well tolerated, with reversible reduction in food intake, with no evidence of degenerative changes or cell injury, no impact on kidney, and they only observed the well-known microvacuolation of cells in the renal cortical tubules and in the urothelium of the urinary tract. Moreover, few cases on the use of intravenous products with high doses of HPBCD and SB-beta-CD in neonates and young children have been reported without signs of toxicity. And this effect of microvacuolation at the renal level, induced by high level of hydroxypropyl or sulfobutyl ether cyclodextrin using intravenous route, can be considered as osmotic nephrosis. And this effect was comparable with intravenous administration of hypertonic sugar, dextran solution, or plasma expander or contrast media, for example. In general, concerning the systemic toxicity, hydroxypropyl or sulfobutyl cyclodextrin induce no toxic effect due to metabolism, no metabolic pathway disturbance, no binding to receptor, no gene expression enhancement, and so on, with no, for example, phase 1 stimulation, no oxidoreduction, hydrolysis with cytochrome, or phase 2 with conjugation with glutathione transferase, sulfoconjugation, no metabolism, or no linkage to plasmatic protein, and so on. Now, concerning ophthalmic or ocular safety of use, in general, cyclodextrin enhances drug penetration into the eyes, but concentration of 4% of alpha cyclodextrin and 5% of randomly methylated cyclodextrin can be toxic to the corneal epithelium of rabbits. Nevertheless, solution of 10% of sulfobutyl ether cyclodextrin and 12% of HPBCD were not found to be toxic or irritating in rabbit eyes. Now, concerning rectal safety of use, cyclodextrin can act as a rectal absorption enhancer of drugs, and cyclodextrin can be absorbed when they are at high percentage in solution. For example, in rats, up to 5% of beta-CD and 26% of HPBCD can be absorbed. And regarding safety at rectal level, suppositories with up to 230 mg of beta-CD and 12% of HPBCD do not cause irritation in the rectal mucosa in human and rabbit respectively. However, alpha cyclodextrin can potentially cause damage to the epithelial cell layer. Now, concerning topical or dermal safety of use of cyclodextrin, cyclodextrin alone are poorly absorbed transdermally, but in combination with absorption-promoting agents, they are able to permeate the skin by 12, 43, and 53% for beta, RAMEB, and HPBCD respectively. Besides, concentration up to 0.1% of alpha, beta, and gamma cyclodextrin are considered safe. Moreover, studies on sensitization, mutagenicity, and topical irritation have proved that HPBCD and beta-CD are safe. In addition, cyclodextrin are currently being used in perfume and cosmetic. Now, concerning nasal safety of use, cyclodextrin are poorly absorbed via mucosal membranes, and at high doses they increased nasal and pulmonary drug permeability by direct action on mucosal membranes, and they can facilitate also their own absorption. Also, they strongly can potentiate lipophilic absorption enhancer, and less than 10% HPBCD or RAMEP solution and less than 1.5 beta-CD solution do not induce tissues damage. Nevertheless, they can cause damage in rats and can keep the integrity of the nasal mucosa. Concerning genotoxicity and mutagenicity aspects, a lot of studies using standard battery of genotoxicity and mutagenicity tests has been performed with alpha, beta, gamma cyclodextrin or HPBCD or SBA cyclodextrin, Ames test, mouse lymphoma, DNA damage, chromosomal aberration test, and so on. And all these studies show that cyclodextrin are non-genotoxic or mutagenic. Now, if we consider the developmental effects of cyclodextrin, some effects were observed for very high dose of beta-CD on rat neonatal growth retardation, with no adverse effect level about 1.25% in the diet. At high doses of HPBCD and SBU-CD, maternal body weight and food consumption were decreased, suggesting maternal toxicity. Moreover, no teratogenic effects were observed following treatment with HPBCD and SBU-beta-CD. Hydroxypropyl beta-cyclodextrin at the highest tested dose level, using 5 g per kilo oral route and 400 mg per kilo intravenous administration, induces a decrease in the survival of the pup at birth and during the early lactation period. And similar results were observed for SBU-beta-CD after intravenous injection of 3 g per kilo. And all these effects may be associated with nutritional deficiency as a consequence of maternal toxicity. And finally, we consider that cyclodextrin are not considered as embryotoxic or teratogenic, and the effects occur at very high doses and were due to maternal toxicity. Then, some words about the regulatory aspects. Concerning the regulatory aspects of cyclodextrin's usage in pharmaceutical applications or in food, both alpha-cyclodextrin alpha-dex and beta-cyclodextrin beta-dex are listed in the European Pharmacopoeia, USP, Chinese Pharmacopoeia, and India Pharmacopoeia. And gamma-cyclodextrin is listed in both USP and Chinese Pharmacopoeia. A monograph for HPBCD is available in the European Pharmacopoeia. In 2000 and 2004, alpha, beta, and gamma cyclodextrin were introduced into generally recognized as safe list of the Food and Drug Administration for use as a food additive. And alpha and beta cyclodextrin are approved as novel food ingredients by the Commission. Beta-CD is approved in Europe as a food additive E459 with an acceptable daily intake of 5 mg per kilo per day. Sulfobutylated cyclodextrin and HPBCD are cited in the FDA's list of inactive pharmaceutical ingredients. Concerning native cyclodextrin monograph, alpha, beta, and gamma cyclodextrin monograph exist in US and Chinese Pharmacopoeia, while only alpha and beta monograph are described in European, Japanese GPE, and Indian Pharmacopoeia. Moreover, the gamma cyclodextrin is only described in US and Chinese Pharmacopoeia. Concerning the Japanese Pharmacopoeia, it deals with API that can also be used as excipients. However, Japan Pharmacopoeia for excipient, GPA, is only for excipients. And this is the reason why no native cyclodextrin are described in Japanese Pharmacopoeia. And concerning the modified cyclodextrin, the most used are HPBCD and to a less extent sulfobutyl beta-CD due to patent reasons, and they have monograph at European and US Pharmacopoeia. In addition, HPBCD has a monograph at China's Pharmacopoeia. Finally, to conclude on these safety aspects of cyclodextrin, cyclodextrin have poor oral biodisponibility. After intravenous administration of cyclodextrins, they disappear rapidly from systemic circulation and are renally excreted intact. Alpha and beta cyclodextrin and RAMEB show more renal toxicity at relatively low doses after parenteral administration, and thus are not suitable for medicinal products given intravenously. Hydroxypropyl beta-cyclodextrin and sulfobutylated beta-cyclodextrin at high doses can cause vacuolation of the kidney tubular cells without loss of kidney function in animals, and this transient increase in size of apical vacuoles is an adaptive response to the excretion of osmotic agents such as glucose, mannitol, and dextran at extremely high concentrations. HPBCD and SBOCD are considered safe at relatively high doses and used most widely in parenteral products. Cyclodextrin are non-mutagenic, genotoxic, carcinogenic, reprotoxic, or teratogene. And, at least, the choice of the most appropriate cyclodextrins in terms of safety has to be done regarding expected use, dermal, oral, rectal, ocular, and so on and so on. And, as always for pharmaceutical applications, the risk assessment must be compared to the effectiveness of the cyclodextrin used, whether as an excipient or as an active ingredient, considering the benefit-risk ratio. So thank you for your attention. If you have any question, feel free.
Thank you very much, Damien, for your very nice presentation. We have time for one question, so I will ask it to you. Would sublingual administration have similar permeability of cyclodextrins as nasal administration?
I think yes, because the mucosal absorption of the cyclodextrin is in fact poor, and we can consider that for the nasal mucosa, it's not very different when we compare in the mouth or sublingual. Maybe we can have some difference at some parts of the nasal route, especially in the contact with the nerves. It can change because of the structure of the cells at this point. But in general, we can consider it could be the same.
Okay, thank you very much, Damien, for your answer.
Thank you.
So let's start now the last but not least presentation of today, titled Next Generation of Hydroxypropyl beta-cyclodextrin as Therapeutics. For this presentation, I'm glad to welcome Tamas Sohajda. Tamas is the CEO of CarboHyde, and he has more than 15 years of history with cyclodextrins. So Tamas, please, the floor is yours.
Okay, hello everyone. My name is Tamas Sohajda, and in my presentation I'll be talking about the next generation of cyclodextrins that are currently used as therapeutics. In my presentation, I'll try to give you a comprehensive overview of all the aspects and all the applications that are currently in the development pipeline, from commercial products to products that are in clinical development, on the way through the most advanced or the major preclinical projects that we see. This is an arbitrary grouping or a differentiation among the cyclodextrins. As I see, there are two main types of applications out there. One is where the cyclodextrins are used as kind of antidotes, and in the other case, cyclodextrins are used to target some disease or symptom where cholesterol homeostasis is influenced. And in the very end of my presentation, I'll talk about the two different concepts chemically, because some groups are using single isomer cyclodextrins, and others are using random derivatives, and I'll try to bring some pros and contras for each concept. So this is just a summary slide about how cyclodextrins are used today. And as I mentioned, we'll be talking about uses as active ingredients. But as you can see, beyond the traditional applications, there are many uses out there in the field of drug delivery and other uses of pharmaceutical applications. So how this all started? This all started with a researcher named Joseph Pita, and a rare indication where there was an issue with the accumulation of vitamin A in the body of a kid, which was treated by the 2,6-dimethylbeta-cyclodextrin. And this was the first application where cyclodextrins was used as actives. As you can see, it was quite a while ago, so actually exactly 30 years ago, where this treatment was introduced, and it was successful in this individual case. So it was not an official treatment. It was a one-time case, but it served as a very cool and valid proof of concept for the pharmaceutical applications of the cyclodextrins. So in this intoxication case, the cyclodextrins proved to be suitable, and this opened the way to a lot of new applications that we are enjoying and researching these days. So the second one, of course, you all know very well, which is Sugamadex, or as the brand name goes, Bridium, which was developed actually by quite a few companies, as you can see on the left bottom. Initially developed by Schering-Plough and AkzoNobel, later took over by Organon, and currently it's marketed by Merck or MSD. Bridion is a blockbuster drug nowadays, and it's been on the market, well, depending on geography, since 2008. Nowadays, its original patents are expiring, which gives rise to a lot of new companies and manufacturers, both on the active ingredient as well as the finished dosage form side. Here on the bottom right corner, you can see the Bridion sales and the forecasts, as well as the great number of companies who tried to develop generic versions of Bridion or came up with alternative technologies for the synthesis, the formulation, and so on. In general, this is a very simple product and concept where the Sugamadex, which was systematically designed cyclodextrin, is used to encapsulate rocuronium and similar neuromuscular blocking agents with similar structures to antagonize its effect and give rise to a very quick reverse of the neuromuscular blockade. Another application where cyclodextrins are used as antidotes, we could say, are in the case of Stargardt disease, which is a rare disease where the accumulation of lipofuscin takes place in the eye, which then leads to symptoms similar to macular degeneration. And this application of cyclodextrins was invented at the Weill Cornell Medicinal College, and then later licensed by Revision Therapeutics, who partnered with Aquis Pharmaceuticals with the intention to start the clinical trials soon. In this case, the cyclodextrin is the commonly used excipient sulfabutyl beta cyclodextrin. At least that's the first lead compound. But there are, of course, ongoing studies supporting that other cyclodextrins could be used for this particular application too. Then from Europe, we also have a promising development which originated from Oklo Life Sciences and then licensed by Alder and Pharma, where the companies are developing a cyclodextrin which reverses the anticoagulant effects of oral anticoagulants. and other platelet inhibitors, as you can see. This project is also in its preclinical phase. Here, the exact structure of the compound is still unknown. Yet the development is supported by some non-dilutive fundings, and it's hopefully entering the clinical phase also soon. So it's late preclinical stage development. And on the wave of the success of Sugamadex, there are more companies and groups who try to develop follow-ups for the compounds with different properties. The two pioneers in this field who we could name are Merck, which is a Chinese company, and the Calgary University, who have different concepts, as you can see, to create Sugamadex follow-ups. They have very similar behavior to Sugamadex, also, as you can see, to some extent similar structures, and they both showed good potency to reverse the effect of the same compounds technically. So rocuronium and structure-related neuromuscular blocking agents. Merck already completed a few phases of clinical trials, and as far as I know, we'll market the product soon in China with very similar therapeutic effects compared to Sugamadex, whereas the compound of Calgary University is still in the preclinical stage. And now with this, I'd like to move on to the compounds which target cholesterol homeostasis. I put the question mark in the bracket for cholesterol targeting, as in this case, the mechanism of action of cyclodextrins is not unambiguously proven. So what we know is that they certainly affect the cholesterol homeostasis. But what we don't know is how this is exactly happening, and there are multiple theories for that, and it's not even excluded that many of them can be true simultaneously. So this can be a direct inclusion complex formation with cholesterol. This can be manifested via some enzymatic activation. This can be manifested by triggering autophagy, and there are other potential explanations how this is done. Some companies even claim that it's not necessarily related to cholesterol, and influence in the cholesterol homeostasis is just a side effect or a byproduct, we could say, of the effect of the cyclodextrins. So I would not really focus on the mechanism of action here, just stating that it's very critical eventually to understand what and how what the cyclodextrin is doing and how it's exerting its effect, in order to, in the future, rationally design new cyclodextrins. Because, as you can see in this case, the currently leading assets originate from an accidental discovery where hydroxypropyl beta cyclodextrin originally was used as an excipient in a formulation, and the vehicle, which contained only cyclodextrin, proved to be efficient, actually more efficient than the active formulation itself. And this is how it was discovered that in the Niemann-Pick C disease, cyclodextrins could indeed be used as therapeutic agents. And from this discovery, eventually two companies with similar yet still different directions spun out. One project has undergone through a number of companies. First, it was developed by Vitas in early phases of clinics, then took over by Sucampo Mallinckrodt, and currently is controlled by a company called Mandos. And the other development is controlled and developed by Cyclotherapeutics. In both cases, the active ingredient is hydroxypropyl beta cyclodextrin, not necessarily the same hydroxypropyl beta cyclodextrin, as we will learn later, but certainly if we call it by this name, it could be used, we could be understood as very similar compounds. So, and in this case, Cyclotherapeutics is the only one who still has active clinical programs and is progressing the development aggressively. They're in late stage of clinics with the Niemann-Pick type C asset. This discovery of cyclodextrins influencing the cholesterol homeostasis triggered a lot of subsequent promising research areas, where always hydroxypropyl beta cyclodextrin was the lead candidate, but therapeutic applications differed a lot. So it was evaluated in a lot of different diseases where cholesterol homeostasis was involved. And in the following examples, we'll tell you more about this. So Cyclotherapeutics has another program which has started its clinical phase just recently, and it's for Alzheimer's disease. There is a company from Luxembourg called Organotherapeutics who reported on the promising application of HPPCD in Parkinson's disease. There is a company in stealth mode in the United States called Baran Therapeutics, or there's a typo there, it's Baran Therapeutics, which uses HPPCD in familiar hypercholesterolemia. There was a nice paper in Nature a few years ago by Sydney University where cyclodextrins were used for the treatment of pox jellyfish poisoning, and I just didn't bring this in the antidote section because the mechanism of action was directly related to the cyclodextrin interacting with cholesterol in the lipid rafts, and thereby it was proven that the effect of ameliorating the symptoms takes place. And there's another company called Zyversa Therapeutics, also from the US, who develops HPPCD for FSGS, which is a kidney disease. So in all these cases, the same cyclodextrin, or the same family of cyclodextrins, hydroxypropyl beta cyclodextrins, are used in the therapy. Of course, there are companies who thought it a bit further and then started evaluating other cyclodextrins in similar indications, we could say. And the first of these companies who really introduced a systematic drug discovery process, and we could say designed cyclodextrin for its target molecule, of course not the first was Sugammadex, but with a similar concept, was Cyclority Therapeutics, who designed a cyclodextrin dimer as their lead candidate, which would target not cholesterol but 7-ketocholesterol, and they have proven via different molecular modeling, so in silico and in vitro techniques, that this interaction is very selective towards this target molecule. And now Cyclority Therapeutics is soon concluding their preclinical developments and initiating the clinical application of their dimers in atherosclerosis. Then there were a lot of other companies, as you could say, who evaluated different cyclodextrins, so cyclodextrins other than HPBCD, but in Niemann-Pick C, and claimed either different mechanism of action or different safety profile or different pharmacokinetics, because eventually we need to admit that cyclodextrins which are used as excipients are not really drug-like compounds. So they get excreted from the body very rapidly, with a half-life of an hour or so. They're not really specific to any organs, not targeting any organs. Of course, they accumulate in higher amounts in the kidney due to their excretion profile, but in general, they're mainly inert to the body. You need high doses of the cyclodextrin. They cannot really permeate any kind of biological barriers. Their bioavailability is close to zero, and similar disadvantages like that, which when you think about them as excipients are really cool, because you can give high doses of them and they're safe and so on. But if you want to use them as active ingredients, they certainly are drawbacks. So what kind of cyclodextrins were used in these studies? Without going into details, hydroxypropyl alpha and gamma cyclodextrins for different concepts. There were sugar-appended cyclodextrins, which were used mainly to target certain organs, so targeted delivery for the active cargo, polyrotaxanes for longer residence time in the body, as well as higher permeations through biological membranes. Peptide cyclodextrin conjugates were also evaluated to target certain tissues and mainly for brain penetration. And cyclodextrin polymers are also used and proposed for these applications, which can stay in the body much longer than the monomers themselves. So if we go beyond these derivatives and these applications, what are out there with the other types of cyclodextrins? So there is a Canadian company called Sol Aeromed, which also uses the invention of Oklo, whom you may recall from a former slide, where they use the common random methyl cyclodextrin, also used as an excipient in different pulmonary inflammatory diseases like cystic fibrosis and asthma, where the cyclodextrin is claimed to have an action via supporting the surfactant function of the lung tissues. It's still in preclinical development and mainly on hold. Another interesting application of cyclodextrins, also mainly methyl cyclodextrins, is in the field of anti-cancer research, dominated by the Japanese Kumamoto University, but we can also find some papers from other, mainly academic institutions, where the cyclodextrin is claimed to trigger apoptosis and lower cell proliferation rate, also again related to cholesterol homeostasis. We should maybe mention that methyl beta cyclodextrin has a different safety profile and some toxic effects compared to other cyclodextrins mentioned in this presentation, but this is certainly an interesting application. And last but not least, especially in the light of the recent events, the antiviral applications of cyclodextrins, where I dedicate a few slides, since they can be distributed into two main groups of cyclodextrins. One is the methyl cyclodextrin again, and the other are the sulfated cyclodextrins, which are used in this field. So how does the antiviral activity of cyclodextrins work? This was maybe one of the first therapeutic applications of cyclodextrins, and the antiviral activity was tested against a lot of different viruses. As you can see, HIV, herpes, influenza, Zika, and so on. There are at least a dozen viruses where cyclodextrins showed some activity. And the mechanism of action was also a bit complicated, or seems to be a bit complicated, because they contribute to the inhibition of the viral entry, also the viral replication. There are also, of course, cholesterol-related effects. In case of the sulfated cyclodextrins, there is a heparinoid-like effect as well, and there is also the autophagy induction. So this kind of antiviral effect can take place on several levels for these cyclodextrins, but what we can say is that there are two main types which are used here, the methyls and the sulfated. Of course, during COVID, this got kind of a hype, and a lot of development was going in this field. Methyl cyclodextrins was first, especially Rocket's CRISMAP, evaluated in this field, but beyond patenting, we couldn't see any human data. unlike sulfated cyclodextrins, where even a company was formed for the development, which is currently running under the name of Esterivir, and as you can see, they have lead candidates against a long list of viruses under development. This was not only limited to pharmaceutical applications, so the antiviral properties of the cyclodextrins were also exploited in, I could say, supporting or preventive products. And one of these was a mouse rinse, which also contained a cyclodextrin to contribute to the antiviral effect, or at least the prevention of the infection. And this is something which was marketed, proven at the smaller clinical level to be beneficial in the prevention. And well, I'm not sure how successful it was, but certainly it has some rationale behind. And now, very quickly about the two different types, single isomer cyclodextrins and random cyclodextrins. On this slide, you can see a few single isomers which are under development. What are the main advantages here? We have a well-defined chemical entity which can be explained very well to regulatories. You can develop analytical methods fairly easily, or at least straightforward. To this, you can follow them pharmacokinetically. You can have an easier way to explain how they are working, track them in vivo. The downsides are that they are usually much more difficult to make, multiple step synthesis involving organic solvents, and you need to develop analytical methods for each step, and eventually they're going to cost much more, especially if chromatography is involved as well. On the other end, well, this is just an example of how an impurity profile for a single isomer cyclodextrin might look like. Shugammadex has over 20, or in some sources 30 reported impurities. So it's very complicated to make and to characterize. For random derivatives, we could say that the synthesis is usually easy and cheap, also water-based, so it's a green technology, and usually they are well established toxicity-wise or safety-wise as excipients for a long time. On the other hand, we are coping with hundreds of components. You cannot really explain which contributes to the efficacy, which contributes to the side effect, which is just balanced into the body completely unnecessary for the therapy. There could be difficulties with batch-to-batch reproducibility and explaining eventually the lack of proper characterization to the regulatories. The analytical methods could be challenging, especially if you want to give them in high detail, and eventually you will not be able to conclude the metabolism, the mechanism of action, the degradation, because you'll always cope with hundreds of components. So what are the random derivatives that we usually know? The captazole, the HPBCD, so kleptos, the methylated cyclodextrins like from Walker, and even cyclaritis dimers are random derivatives. Even with random derivatives, the proper characterization is necessary, and when we use sophisticated analytical tools, you can see that, for instance, two different hydroxypropyl beta cyclodextrins are indeed very different because their composition is pretty much different. And this is just a glimpse of what single isomer cyclodextrin look like. So it's not even straightforward which single isomer you use, because we're talking about three potential positions that you can substitute on the cyclodextrin, not to talk about the different number of glucose units. So if you are considering a single isomer cyclodextrin, you're eventually considering a lot of options and a lot of potential impurities or byproducts. So what is the take-home message of all these huge amount of information? That there is a new era coming for cyclodextrins, which is flooded by uses as therapeutic agents and novel drug delivery systems, and those mainly include new cyclodextrins. And then there is a real challenge in these innovative areas, because we now know that cyclodextrins that we have been using for a long time can be used in these indications, but we can also think that these were not essentially designed for such applications. And if we design systematically new cyclodextrins, knowing how they are working and what they are doing with the body, we can come up with much better derivatives. I should also mention that since Sugambadax, now new cyclodextrin was introduced to the pharmaceutical market, which is now almost 20 years. So there is a high time for such new compounds to emerge. And in order to do that, there are two main challenges that should be overcome: the understanding of the mechanism of action, and then the proper design of new cyclodextrins. And with that, I'd like to thank you for your attention, and I hope that you have a lot of questions for my talk. Thank you.
Thank you very much, Tamas, for your very nice presentation. We have time for one question. In your opinion, which holds the future in the therapeutics use of cyclodextrins, novel derivatives or well-established compounds?
Well, I would say that that the cyclodextrins that are on the shelf are very good models to evaluate potential uses for new indications, but eventually I put my vote to new compounds, because I think, in my opinion, that's the only way to really create molecules that are efficient. And currently, that's one of the main drawbacks of therapeutic applications, that you have to give really high doses to the patients, which are, from one point of view, due to the lack of efficacy, I mean high efficacy levels, and the other point of view, to the lack of proper pharmacokinetics. And those could only be overcome by compounds which possess different physicochemical properties than the current molecules. And before I give the word back to you, I have to make one correction about one of the programs I talked about, over Pharmaceuticals, because they just came up with a new press release a week ago that they just concluded their phase 1 clinical trial. So the program of Alberon and Oclo has passed the first in human trials for rapid treatment of intracranial hemorrhage. So that's another and certainly a new cyclodextrin which is on the way to the market, which is very good news.
Okay, thank you, Tamas, for your answer and for this slight correction. Now it's time to open the panel session. So I'm very glad to welcome Professor Loftsson from the Faculty of Pharmaceutical Sciences of the University of Iceland, and Professor Bilansoy from the Faculty of Pharmacy of ASCTP University in Turkey. So first of all, first question maybe to Professor Loftsson. How do you see the future of cyclodextrins in the pharma world?
Well, I think it's just about starting. We can see that early on we had this, you would say, revolution in parenteral formulation, where we were able to get basically rid of organic solvents and surfactants and replace it with carbohydrates, which are very rapidly eliminated from the systemic blood circulation. So I think it is a revolution, you could say, in the excipients, for example, for the parenteral formulations. Also, all the formulations have this tendency to give precipitates when you inject, and cyclodextrin don't have this tendency. It's usually, you don't see a precipitation after injection. That is just one. In oral formulation, we are seeing something similar. The only thing there, we have this problem of formulation bulk, because we have this heavy molecular weight. They are heavy, so we are increasing the size of the solid dosage forms for oral delivery. So that is a problem. But otherwise, it has been giving us a very good results, good bioavailability, and so on. Then for other types of formulations, intranasal, ophthalmic, and so on, buccal, it has a great potency, and I think it's just starting in these areas. Yeah.
Thank you, Professor Lofton. Professor Bilansoy, on your side, for the same question, how do you see the future of cyclodextrins in pharma?
Well, thank you. I actually would like to respond to this question depending on my experience with cyclodextrins. So I have been working with cyclodextrins since my PhD, and mostly I have been using amphiphilic cyclodextrins as drug delivery systems and carriers in formulations of some active pharmaceutical ingredients with bioavailability problems, either because of their solubility problem or because they are physiologically unstable. So what we came through in our studies were basically the potential of these amphiphilic cyclodextrins to form spontaneous nanoparticles. So they were able to both deliver these problematic drugs to target sites, which were in my studies either tumors or inflammation sites. They were also able to selectively kill cancer cells, which we believe were a result of their affinity to the membrane cholesterol and their ability to extract this membrane cholesterol, leading the cell to apoptosis. And also we were able to, apart from the nanotechnological approach, we were able to incorporate some of the cyclodextrins like HP beta CD, for example, to 3D printing application of pharmaceuticals, where these cyclodextrins were used to stabilize the active ingredient and inhibit recrystallization during the printing process when the drug is in the filament or in the bio-ink. So in my opinion, I think I agree with Tamas here. I think that innovative cyclodextrins hold a great potential for the future. But of course, the potential of cyclodextrins that are already defined in pharmacopoeial monographs and are described in EMA guidelines are valid in use in pharmaceutical formulations. So these new derivatives need to be characterized thoroughly in terms of both quality, reproducibility, scale-up, efficacy, therapeutic efficacy, and also, of course, safety, which is always a big problem depending on your administration route. So we have experience with oral and parenteral administration of these new amphiphilic cyclodextrins. And I think that especially the cationic cyclodextrins hold a considerable potential for oral and mucosal application. And I think this is basically what I would like to underline at this stage.
Okay, thank you. So my second question was regarding the regulatory and safety aspect. Do you think that they can hurdle the development of novel cyclodextrins?
In terms of regulations?
Yeah.
The amphiphilic cyclodextrins, they really have to, study it very well clinically. At this point in time, we don't have that clinical data in hand. We just have animal data and cell culture data. So both acute and systemic toxicity profiles should be well established, together with dose response profiles as well. So there is a long way in becoming regulatory safe for these products, I think.
Yeah, Professor Lofton, on your side, do you think that regulatory and safety aspect can be hurdles in developing novel cyclodextrins?
I think, like Tamas said, Tamas was mainly talking about biologically active cyclodextrin, which can be useful. And I think RM was also talking about that. For excipients, it will be difficult. It has always been difficult to market new excipients because you need to take a patent on it, and the patent you will have 20, 21 years, And from marketing point of view, it will take you longer time to market it, and so you will not have this protection. It's very difficult. For excipients, I think you will try to stick with current cyclodextrin therapeutics. You might be able to develop new cyclodextrin derivatives, new cyclodextrin as excipients, but they have to have some special properties which the other current cyclodextrin don't have. And I think that will be difficult. But as new chemical entities for new therapeutic applications, I think like Eren and Tamas were saying, new cyclodextrin have a great potential. And this is totally different from excipients. It's a different development, you could say.
Okay, thank you. And therefore, how do you see the future in developing novel modified cyclodextrin or hydroxypropylated beta cyclodextrin as effective drug in selected or targeted diseases?
I think it is very interesting. I've not been active in this area myself, but I've seen this activity. It's also very nice to see that even the cyclodextrin, like hydroxypropyl beta cyclodextrin and other cyclodextrins, they have a certain biological activity. We tend not to talk too much about it, because if we talk about it, then we can get into difficulties with the therapeutic, with the regulatory authorities. If we start to say that hydroxypropyl beta cyclodextrin has a biological activity, the authorities will have different approach and start to ask you questions about it. So people tend to keep their mouth shut. But we have seen it in eye drops and other things, that the cyclodextrin formulations are very positive. They have this very, very nice feeling when you have it, you give them in an eye drop to the eye, to humans. And even we have seen that they reduce irritation of lots of drugs. And it's not only because they are protecting the eye from the drug, which they might do, but also I think the cyclodextrins themselves have irritation reducing activity. So we know people like me, which have been working with cyclodextrins over the year, we know that they have positive effect. They are very, very good, and most probably they have a mild reducing, irritating, reducing, anti-inflammatory effect, and so on. But we tend not to talk about it because of regulatory issues. We never can claim it, you know. When you formulate the drug, you have to list why are you using cyclodextrin, why are you using this polymer, and so on. And you claim it, and you always claim the cyclodextrins as a solubilizer. You cannot claim it to reduce irritation. I don't think. Yes, you can claim it, but not as an active. You cannot do that. And if you do that, you can get into trouble. So it's a strange thing, you know, what we do. I see Tamas is laughing at me probably. But this is the situation. This is how things are. Yeah.
Thank you. And on your side, Professor Belançoy, how do you see the future in developing novel modified cyclodextrins for selected or targeted diseases?
Yeah, I think I agree with Professor Loftsson that in terms of excipients or biologically active ingredients, the cyclodextrins have a major problem in terms of their purity, reproducibility, and batch-to-batch variability. I think the rate-limiting step here is the well-established synthesis and characterization of the final product. that is going to be marketed as an excipient or as an active ingredient in terms of a new cyclodextrin. Now, I have synthesized the cyclodextrins that I worked with, and I know how difficult it is to purify the new cyclodextrin and crystallize it. So I respect chemists enormously, but I think in large scale, I think this is one of the major problems, because since there is statistical substitution, the risk of statistical substitution, reproducibility of the same product can be a challenge. And in a pharmaceutical product, in terms of regulatory approach and also in terms of safety, and this is a major challenge to be overcome. So I think, in my opinion, the major problem here is this, but there are also, of course, promising steps in the use of cyclodextrins in the COVID vaccines and other parenteral products for chronic diseases, which require repeated administration of this cyclodextrin. So I think chemists should be working on this problem. I know that they already do, but I think this is the most important obstacle for the cyclodextrins, to have a wider scale presence in the pharmaceutical market.
Yeah, I would like to add to this, it's one thing we have observed also working with cyclodextrin from different manufacturers, is that sometimes they are expensive, you think that you can maybe buy them from China or some other unknown supplier, and then you frequently get into trouble, both because of the purity of the cyclodextrins you get, and also because they appear to have different properties, different quality, probably because of different distribution of the substituents or other things. So I think for the audience, for people, I think they should buy the cyclodextrin from a well-known supplier like Rocket or Wacker or someone else, which we know are known for their good quality cyclodextrin. They can try cyclodextrin from other sources, but they should be careful about it and should be open-minded, think about the quality of the products they are getting from those sources.
Thank you very much. Tamas, do you want to comment on the development of novel modified cyclodextrin for selected or targeted diseases?
Yeah, maybe just I would add that it's not only the batch-to-batch reproducibility, but if you see any differences in the performance or the functional properties of the different cyclodextrins in lack of currently at least the proper analytical tools that enable very high level understanding of the composition, you will not be able to explain why they are working differently. I've seen that in the past that companies use cyclodextrins that according to the valid analytical methods seem to be the same, and they were performing differently. That goes, of course, to random derivatives, and it was not possible or very unlikely to explain what is the real difference between the two, at least following the official methods. So and that could also be avoided by the use of single isomer cyclodextrins, where the characterization is more straightforward at least. But otherwise, I of course agree with the professors. Very good points.
Thank you, Tamas. Regarding the future of modified cyclodextrin, what is your opinion about the modification like grafted cyclodextrin with sugars or peptides or other type of molecules? Maybe first Professor Bilansoy.
Until now, I have a lot of experience with cyclodextrins modified with aliphatic chains to render an amphiphilic property so that the cyclodextrin will be able to form nanoparticles spontaneously. I think that these molecules relatively simpler to synthesize and have a potential because of their ability to extract the cholesterol, especially the membrane cholesterol. I would really love to see how they have an effect on hyperlipidemia, how they have an effect on obesity, for example. I think they have a good potential as a nanoparticle forming agent. Recently, I have 2 different projects where we are conjugating a monoclonal antibody to the cyclodextrin, to the amphiphilic cyclodextrin nanoparticle, so that we can actively target some tumor types. And this works quite well again. So in my opinion and in my experience, I would be focusing on these different modified cyclodextrins for future applications. Regarding the fact that we have more and more nanomedicines in the market, there are now more than 80 different nanomedicines, and there are also, of course, generic nanomedicines called nanosimilars. So I think that cyclodextrins have this potential to be used in the formulation of these nanomedicines and their similars as well. But of course, I don't have experience with other sugar modified or peptide modified cyclodextrins. So I would leave the microphone to Professor Lofton in that case.
Well, I would like to mention that cyclodextrins themselves, they form nanoparticles, and they are able to form microparticles, and that will affect their type of delivery, how they deliver drugs. But these particles are maybe different because they are not very stable, they fall apart, which is good in a way, but also it has these drawbacks, but it has also some positive effects because of toxicology and other things. And other thing which I would mention, which I noticed during the lecture, that it was mentioned that alpha cyclodextrin is maybe not used in oral formulations. I think it was in the list there. I would like to mention that there is a product on the market in America or Canada, at least. I think it's also in the US, where the daily dose is about, I think it was about 600 milligrams of alpha cyclodextrin used. It came to me, it's used for weight reduction, actually, because it is supposed or does complex triglycerides and prevents absorption of triglycerides into the body. So that is also when people are using just the natural cyclodextrin as a weight regulatory, reduce the weight. So that is also there. So yes, I can understand that people are looking for new cyclodextrin derivatives. I think it's a very exciting topic, but I think we have lots to do also with the current cyclodextrin, the natural cyclodextrin as they are today, alpha, beta, gamma, and the hydroxypropyl beta, and the sulfobutyl ether beta cyclodextrin, and so on. They will be very useful for many, many years to come as pharmaceutical excipients. But new cyclodextrin derivatives, yes, they will of course change a lot, and they will have biological activities or could be good for some site-specific drug delivery to tumors or some other tissues within the body.
Thank you. So very optimistic future for cyclodextrins. What are the main focus of academic researchers on cyclodextrin today? Can you elaborate on that?
Okay. There are twofold, you know. There is a lot of activity on new cyclodextrin derivatives, cyclodextrin polymers and cyclodextrin derivatives with specific properties. But then we have lots of studies on the applied science, with application of cyclodextrin to help us to deliver drugs into the body. And cyclodextrins, in the early days, we used organic solvents to do the tox studies, the first tox studies of new chemical entities in animals when we wanted to test them in animals. Nowadays, more and more companies are using aqueous cyclodextrin solutions because the cyclodextrin has such a low toxicity, much lower than organic solvents, and you can use pure aqueous solutions with cyclodextrin instead of organic solvents or surfactants. So that is very, very positive. But again, well, I think we will continue to explore, and I think it will be more exploratory, more into people that just use them in their studies, in other academic studies. But I think within the cyclodextrin field itself, you can see it in like the European cyclodextrin meeting in Budapest recently. If you look at the presentations, they were mainly about new chemical entities, new cyclodextrin derivatives.
And what about your point of view, Professor Bilansou, on the main focus of academic research on cyclodextrins today?
Yes, of course, the academic research on cyclodextrins is based on the basic properties of cyclodextrins in terms of forming inclusion complexes, and also because of their ability to pass membranes, biological membranes. But I think that in the academic field now, there's growing interest in elucidating the biological effect of the cyclodextrin itself at the molecular level. And I think for this, of course, there are a very wide range of different cyclodextrin derivatives that are being studied, but I think that extensive metabolomic and proteomic studies should be performed on these derivatives that have a good potential for drug delivery or drug formulations, because most of the cyclodextrins exert a biological effect on different cell types. We have come across some cyclodextrins that suppress, for example, cholesterol biosynthesis in the body, some cyclodextrins that facilitate the transport of small molecules through membranes. Some have a suppressive effect on some metabolic pathways, or they induce some other metabolic pathways, and also they overexpress or underexpress certain proteins in these cells. So these effects should be fully elucidated to actually make use of these new cyclodextrin derivatives and really benefit from their potential of therapeutic efficacy, and also be sure about their safety. So I think that the effect, their intrinsic effect in the body should be well elucidated, because actually, if you know the mechanism of some effect in the body, you can work on it, add to it, and come up with new innovative products. So I think maybe this is one of the major focus points of the academic research for cyclodextrins.
Thank you, Professor Bilansou. I would like to ask a question to Carmen. Carmen, do you think there is still room to modify cyclodextrins for enhancing their functionality?
I think, okay, sorry, I don't have video. Can you hear me?
Yes, we can hear you.
Okay, so I agree with all previous opinion, but I think we should start to make a very strong distinction when I talk cyclodextrin as an excipient and cyclodextrin as an API. Of course, if we do new type of cyclodextrins, that will be excellent if we can add new functionalities, but that will be a new excipient if we are in excipient category, and I don't know who is going to pay for all the studies and so on. So you have to work with a big pharma company which is in a crisis, to create a cyclodextrin, which will help their API. So that will be one policy. When we are talking about APIs, cyclodextrin and APIs, we have to be very careful. We are talking generally cholesterol, but there are so many types of cholesterols. We have to look what cholesterol is this cyclodextrin acting on, because if it's acting indiscriminately, that is not good. So that is not a good API. If it's acting on certain type of cholesterol involving certain type of injuries at the cell level, that is a different matter. So the toxicology here will be very important. Secondly, I think we should develop a lot of new methods to physically prove what is happening at the molecular level. Yet we are still have models, but we cannot evaluate exactly what is happening, because here you have a thermodynamic interaction between your actives, the cell, and so on. So everything is not connected like in chemistry with a covalent reaction. There you can say this is the product, what is happening. So I think it's a lot of field of evaluation and toxicology to be first developed, and then we can say, oh, we need new cyclodextrins.
Thank you, Carmen. So we will soon close this webinar. So I would like to give the last word to Professor Lofton and Bill-François. So Professor Lofton, what is your final word regarding cyclodextrin?
I think Carmen had a very good point that she wanted to, if I understood her right, to divide basically this field into two parts: excipients and APIs. And I think how we approach cyclodextrin and how we study cyclodextrin, we have to do it these two different ways. One is as an excipient, it's different. And then as an API, which is totally different, because we have different approaches, different studies are required, and so on. So I think it was very good what Carmen said there about these two, you could say, two phases of cyclodextrins.
Thank you, Professor Lofton. Professor Bill-François, do you have a last word for us?
Yes, I both agree with Carmen and Professor Lofton, and I want to add that there's a synergy about companies and academic centers working on cyclodextrins and coming together in projects or in different conferences dedicated to this molecule family is crucial in moving forward. I want to thank you for this opportunity. It was a very nice webinar, and I hope to continue with working on cyclodextrins with all of you together.
Thank you very much to you, Professor Lofton, Professor Bill-François. Thank you very much to all the attendees. There is a few remaining questions that we have not answered yet. We will answer them writing by later on. I remind you that you will have online access to the recording of this webinar within the next days. So I thank you again very much for your attention, and have a good day. Bye-bye.
Thank you.
Thank you very much.
Thank you very much.
Bye.
Thank you. Bye-bye.
Bye-bye.